Hey everyone, and welcome to week 5 in environmental science. This week we'll be studying content from chapter 8 in our textbook about conservation and biodiversity. This tab in the content page has some learning objectives to help guide your reading of chapter 8. Here's some terms to know, and then I've got some videos and study tables for you to fill out, and just some other support resources to try to make this content come to life for you. Here's another example of a study table and a video. This week's discussion is going to get us digging deeper into the wolf reintroduction that happened in Yellowstone National Park back in the early 2000s. So you already learned a little bit about what happened there in the week 4 content page. This week you're going to dig a little deeper. That was a really simple ecological story about how the ecosystem was impacted by wolf reintroduction. This is a much more complicated look. This is a peer review paper that looked at just the community response. This is a New York Times video that takes a look at sort of the social and political implications of wolf reintroduction. And then here's a couple of articles that describe the Native American perspective about wolf conservation. So these are some resources to help you get to know the topic. By Wednesday of week 5, I'm asking you to make an initial post where you describe why gray wolf protection under the Endangered Species Act is controversial, which of the perspectives in the media above or outside resources resonates most strongly with you. Is it the ecological perspectives? Is it some of the social perspectives? And then I'm asking you to describe whether you think wolf reintroduction in Yellowstone was justified and explain why or why not. Then check back into the discussion later in the week by Saturday and respond to at least 2 of your peers. Make sure that your responses are adding new information to the discussion. So don't just tell them they did a great job. Make sure you really kind of dig in there and offer new insights or maybe information that contradicts what they said or supports what they said. But we need to go beyond just telling them they did a nice job. This week's lab is another outdoor ecology lab. And so you need to make sure that you're not starting to do this lab Saturday night when it's dark. So let's just do it 11:59 p.m. on Saturday, but you will not be successful doing this lab in the dark. So you need to find a safe place where you can sample a 10 by 10 meter area outside. So I know some of you still may have a little bit of snow on the ground. The Weed COS campus has 10 by 10 meter areas that are clear. So if you don't have a spot in your backyard or your local forest, you can come here and do the sampling. It's actually better if you do this in a natural community rather than a landscaped area like your backyard. The details about this lab are on this page. This is a little background about some of the underlying ecology. This is about a community. So the last ecology lab that you did was about a single species. That's a population. A community is defined as all of the species interacting with each other in a defined area. So in this process, you're going to follow a similar sampling technique in terms of the area and the size of the plots sampled and how you randomly select the plots. But instead of just looking at the number of individuals of one species, you're going to be recording the number of individuals of all of the different species that are in that plot. So this is some background information. Here's the field protocol. You should have almost everything you need. If you did get one of our free kits, you should have almost everything you need. You may need some objects to delineate the sampling area, like rocks or cones, something you find out there in the field. You may need some objects like pine cones or forks to show the plot corners for your 1 by 1 meter plots within your 10 by 10 meter sampling area. So this text is going to walk you through what to do. There's also some videos of me locating plots and sampling a first plot and then a second plot. And then here's the sample data sheet that I filled out from those plots. So you can see what that looks like and make sure that you're doing your own data sheet right, which is right here. So you're going to sample 5 plots total following these instructions. When you're done, you're going to complete the table by writing the total number of individuals from species number 1, 2, 3, 4, however many species you found, calculating their relative abundance, calculating the species richness, and calculating the Simpson's index of biodiversity. There's some text to walk you through making those calculations on this page. Here's the sample data sheet that's been filled out. And then the last thing that you're going to do is make a rank abundance curve for your data. This is walking you through how to set it up in Excel and then make the graph. Once you have a graph, I'm asking everybody to go to this class data sharing discussion, post a description of the area you sampled, and then also your rank abundance curve or your graph. When you're done with that, please answer these questions. One of them asks you to look at the class data. So please try to get that up there early in the week so that your peers have some other data to compare theirs to. You will turn all of this in on this page. Here's the grading information. Here's what I'm looking for. Completed data table, your data sharing post, answers to the questions, and your completed species area curve. This is the class discussion where you'll share your data so others can compare theirs to what you found. And then the last 2 pieces of the week 5 module are a practice quiz, a 0-point practice quiz that's there to help you get ready for the 10-point graded quiz. As always, please let me know if you need any help this week. Take care.